So you can see the base. So there's little numbers indicating the base pairs of each of those fragments. So what you would do with this information is plot the distance that it moved on the gel versus the fragment size. And then that gives you a way to calibrate so you can figure out how big your own fragment sizes are. Okay, and then here we have the different combinations. So we have a non-taster would look like that. So we have the undigested DNA and the digested. They both look exactly the same. Taster would look more like this. And then heterozygous taster would look like this. So you'd have two really strong bands there. And again, the undigested band looks similar. It's in the same place for all three of them. And the digested bands are where you'd see the differences. Okay. And then there's another kind of ladder on this side too. I'm not sure why they have two, but it's another ladder you might use for calibration. Okay. So you can see how clear this is. This is why people like to use ethidium bromide, because it glows and it's really easy to see and very clear. And then, so that's what that looks like. Now, let's see if you guys can see this. It might be hard to see because I think when I put the light table on, you're going to get a weird, you get that weird Venetian blind effect. I don't know why. It's something, so to me, this looks just like a pure table of light, but yeah. I think it's because there's a fluorescent light in there, so it does something a bit strange. So, what else can I do here? Maybe I'll turn this off and try to turn. This is the first time we've attempted this over Zoom, so it's we can just blame coronavirus if we can't see anything here. Let's see. Still a bit shiny. I'm going to turn some lights off in here.
Okay. Oh, yeah.
Can you see it, sort of?
Yep. Just see a few bands, yeah.
You can see the ladder.
Yep. Oh, yeah, that's even better.
Hey, Maria, whenever you talk, your screen takes away the image. So I can't see when you talk most of the time.
You have to pin it, Katie.
Wait, what do I have to do?
There's a little pin to the upper corner. Just pin Robyn so that it doesn't move.
Oh, okay.
Yeah, great, thank you, thanks. Great, okay, so let's just zoom out a little bit here. Okay, so this is, oh yeah, okay, that's not bad. It would look more clear if you were here, but yeah, so this is the ladder that we did. So I loaded a ladder there. I also did an extra ladder down on this side. And you can see the little fragments of known sizes. So what you would do is measure from the origin point to each of these fragments, and then plot that measurement against the known sizes of the fragments. And then your little pieces of DNA would be kind of down in here. And as you can see, it's very, I'm just seeing a trace of one of the bands. I think it would be very hard for you guys to see, but they're very light. And that's just an indication that there's not a lot of DNA in there, which tells me somewhere along the line, we didn't get as many copies of it as we had hoped. And there could be a couple reasons for that we'll talk about in a sec. The other thing is, if I leave this to destain a little longer, the background blue will go away a bit more, which will make the bands stand out. So let me show you one from last year that's been destaining for a year, and it'll be, I think, a little more clear. Let's see how this one looks. Oh yeah, there you go. See, you can see those bands there. And this one, even last year, it was the bands were more obvious than in your gel, but you can see right there. Okay, so we have an undigested and digested. In the digested, we can see two bands. Again, it'll be easier if you were here to see it with the light table on. Put that on again and see what it does. Yeah, then you're just getting that weird, oh yeah, you can kind of see them there. Yeah. Okay, so that's kind of the idea. So then, again, you would be able to measure down from the origin point to those bands, and then you'd figure out the size of the different fragments. Okay. And of course, one of the reasons for the ladder, so the distance these travel will kind of depend on how long I left them running on the electrophoresis. So that's why we need this ladder. Every gel needs its own ladder, because if I left this longer, the ladder would travel further, a little further as well. And then that would give you the calibration for your own bands. Okay, one more here from last year that had really strong looking bands on it. Ooh, there we go. Look at that. So I don't even know what this gel was, because it's I have a bunch of gels all in a bin. This one just looks so good that I just don't even know how we got it to look so good. But I don't know whose DNA that was, but this was super successful. So there's a lot of DNA there. So this is, if everything goes perfectly during the PCR process and you get a lot of DNA, then you'll have a stronger looking band, right? Okay. So let's do a little bit of just some troubleshooting together, because this is actually, if you were in a lab and you had to figure this out, this would be how you might do it. So we have this gel, and I ran it, and we're not really seeing much DNA on there. The ladder looks okay. The ladder comes to me pre-prepared. So it's pre-prepared in a lab, and it's all good. So that I just load right into the well, and it's fine. But our own DNA doesn't look that good. So what kinds of things do you think could have gone wrong, or not ideally, to cause this to happen? To have so little DNA that we're not really seeing it, if you think through the procedure. Not necessarily things that we did. Let's think of things that we could have done, but then also things that may have been beyond our control. Any ideas? Okay, I'll tell you then. So, when
I know that you could have accidentally stuck gone too far, gone through the gel, for example, but that's not out of your control.
Yes, that's absolutely true. I could have, if I had injected it too high or too low in the gel, the sample might have just drifted away. That's very much a possibility. In this case, I know that didn't happen because I saw them all nestled in there properly when I loaded it. But yes, that's one thing that could have happened. Anything else?
Do you think that the gel could be too thick or too thin? The gel itself?
Possibly, yeah. It would make it a bit harder to see the DNA if it was too thick. And so there's okay.
There's a second part where, like, this after the second like centrifuge where we added like the primer or something, like you couldn't really see what you were picking up very clearly. Like, so maybe we didn't pick up enough of the sample, or it was off in some way.
Yeah. Okay. So like when you were adding your little bit of DNA back into the primer, yeah, that definitely could have happened. Now, when I think about that, my thought is that all of them aren't there. So that would mean that all of us would have had to do that. So I would think that would be a possibility if maybe one row didn't look that good, but because they all look kind of not like they're not there, that tells me it's something that was common to all of the treatments. So what are some of the other things that were common to all of our treatments? This is some good answers here. Okay. I see Katie shaking her head. I don't know. Okay, so I suspect, actually there's three things that I suspect could have caused less than ideal results. We had the little PCR ball, that little bead that had the polymerase in it. So it's possible, those have a shelf life. So it's possible if it was a little older, maybe it had degraded a little bit and the enzyme wasn't as effective as we would have liked. which would have meant we wouldn't get as many copies of the DNA. And then the other thing is the little reagent that we had, the primer, that as well. If it was sort of close to its shelf life or it was a little off, that also would account for all of them being off. And then the other common thing is the PCR machine itself. So we're just thinking that it's working the way it says it is, but if you were in a professional lab, there would be like a calibration that you might do. So you'd have someone come in and calibrate it to make sure it's actually doing what it says it's doing. So I'm relying on this computer screen that's saying, here's what it's doing, here's the temperatures, but how do I know that's actually happening? And that's why usually in a lab, someone would come and certify it and actually test that it's doing what it's supposed to. So those are some of the things. And I think there could be other things too, but again, I have a second gel, so we'll see what the next gel looks like. Maybe I'll go get it right now and see if we can see anything.
Okay.
It's just right over. I'll be right back.
Awesome. So if the staining doesn't destain enough so that we can see your DNA, we'll use a previous year's. Can we do that, Robyn?
Yeah, definitely. I have a couple of really good ones here. This one just put it on there. Yeah, it's still pretty dark. I don't.
Oh, that's
I think
pretty dark.
Yeah, it needs to destain a little longer. See how? So this is why the destaining is really important as well. So I stained it with methylene blue. When you first take it out, it's really dark blue, and then gradually all the background loses the stain and then the band. So I can see the ladder a little bit. I can see some dark kind of clouds down where your band should be. So I suspect they'll pop out, but I think it needs a bit more time. Okay. So
Okay.
yeah. Okay. So Maria, do you want me to I'll keep destaining these, but in the meantime, should I send you a picture of one of the ones from last year? Or
Yes,
how do
that
you
would be
want me to
great.
transmit? Okay.
I'm wondering how, since we're not, remember when we were in the lab, we actually use the light table and the students measure the distance from the well to the DNA right on the light table in real distance.
Yeah.
If we take a picture, the relative distance will still be the same.
Yes, that's true. Yep.
So
yeah, you're right. You'll still be able to do it from the picture.
yeah.
So
Okay.
I'll take, I'll find the best one and send you guys a picture, and then you'll be able to measure it from the picture.
That would be awesome.
And then I think I'm here for a couple more hours, so I'll look at your gels before I leave to see how they look.
Okay.
And I'll send Maria a picture. If not, I know you probably need this data to start your assignments, but if they're not looking good tonight, by next week they might look better. So you might at least get them as a souvenir or a picture of it as a souvenir. But in that case, you probably want to use the data from this old gel. So
And well, if we all do this gel first, well, everyone will know how to do it.
yes,
So
perfect.
if the students' own gels work out, then they can just replace the data with that.
That
Draw
sounds
their
great.
conclusions. Yeah.
Okay.
Excellent. Thanks.
Good. Okay, so thanks everyone. Thanks for your great questions. Do you have any other questions for me before I let you go? Nope. All right.
No?
Thanks, Robyn.
Yeah, thanks guys.
Yes, thanks a million. Really awesome.
Yeah, thanks. Okay, thanks for all your great work, and I will see you soon.
Okay.
Okay.
Bye for now.